Hello, we are Group 21. We are going to talk about T. T is tumor, I is infiltrating, L is lymphocytes. Before we talk about T's, explain about lymphocytes. Lymphocytes are one type of white blood cells. There are three main types of lymphocytes: B cells, T cells, and natural killer cells. They play an important role of immune system. They recognize foreign substances in the body and try to eliminate them. When cancers appear and grow in the body, lymphocytes recognize them abnormal and migrate toward cancers. These lymphocytes are called tumor infiltrating lymphocytes, or TILs. TILs are the same as lymphocytes, so they consist of B cells, T cells, and natural killer cells. Among these, CD8 positive T cells and CD4 positive T cells account for majority. Natural killer cells are rare and B cells are even rare. Next, I will explain how these two cells and natural killer cells act on cancer cells. First, talk about CD8 positive T cells. T cell receptors on the surface of CD8 positive T cells recognize antigen peptide presented on MHC class I molecules of cancer cells. After recognizing, T cells attack cancer cells. At this time, when PD-L1 of cancer cells binds to PD-1 of T cells, T cell activity is suppressed. Immune checkpoint inhibitors act at this site and prevent T cell suppression. Next, about CD4 positive T cells. There are several types of CD4 positive T cells. Among them, Th1 cells have the function of activating CD8 positive T cells and have anti-tumor effect. However, Treg has the function of suppressing CD8 positive T cells, so it has a positive effect on cancer. Finally, about natural killer cells. MHC class I specific inhibitory on the surface natural killer cells recognize MHC class I molecules of cancer cells. However, unlike T cells, natural killer cells attack cells that do not have MHC class I molecules. TILs are very effective in cancer treatment because they already recognize many targets on the cancer cells. In this method, T cells are removed from a patient's tumor and grow in culture. Cancer cells can then be removed more efficiently by returning the proliferated T cells to the patient's body. Other cancer treatments that use T cells include TCR T cell therapy and CAR T therapy. Both treatments involve genetically modifying T cells. Then, the T cells are made to express receptors that allow them to recognize foreign cells, such as cancer cells. TCR-T expresses a receptor that recognizes cancer antigen peptides, and CAR-T expresses a chimeric antigen receptor that recognizes cancer antigen. The differences in receptor type is the difference between the two treatments. In addition, these two treatments require genetic modification in order for T cells to recognize cancer. But TIL is a more effective treatment because it proliferates T cells that already recognize cancer cells. It is considered. That's all for TIL's introduction. Thank you for listening.